Okay, so now we'll turn to networks. So pathways capture only the well understood portion of biology, things that are understood enough to get it into the scientific literature. And in particular, they don't capture any of the high throughput information like proteomic pull down information, genetic interaction data, RNA co-expression data, and other relationships where we know there's some relationship between genes or RNAs, but we don't know exactly what they are. So networks are more flexible. They can cover less well understood relationships, including those genetic interactions, physical interactions, co-expression, go-term sharing, or concepts like adjacency and pathways. So network databases, unlike pathway databases, can be built automatically by taking large published data sets, going to their supplementary files, downloading them and digesting them. As a result, they have more extensive coverage of biological systems, but the relationships and the underlying evidence that says that two proteins interact with each other, for example, is more tentative because it's coming from high throughput, noisy data. There are many, many network databases. There are over 400 of them. Three of the largest ones are BioGrid, Intact, and GeneMania, and the URLs for all these are in your notes. They have tens of thousands of genes in them. BioGrid has 89,000 genes and over 2 million interactions across 80 species. Intact has 143,000 interactors and 1.5 million interactions. And GeneMania, which is one of Gary Bader's projects, is a compendium of 2,800 networks that together represent 167,000 genes and 660 million interactions. So it is quite large. Here's an example of how you would interact with one of the network databases. This is a screenshot from Intact. I have typed in, I think, TP53 as the example here, and it has pulled out everything that TP53 interacts with, either genetically or physically, in a series of high throughput experiments. P53 is in the middle of this hairball here, and everything that it's interacting with is surrounds it. You can expand this network and show interactors of the interactors, and slowly turn into a big hairball that's very hard to understand, to be frank. GeneMania actually has a similar interface, but it is more interactive and actually a lot of fun to use. You can select the type of network you want to look at. I want to look at genetic networks. I want to look at RNA co-expression networks. I want to look at physical interaction networks, or you can combine them. And then when you do a search for P53 in this example, which is in the middle, it shows you the interactions that are supported by multiple lines of evidence. So you can tune this so that instead of showing you every genetic interaction that's ever been published, it shows you genetic interactions which are supported by other evidence, such as physical interactions. So you can tune the size of this network and increase the amount of evidence for interactions. And it has a very nice feature that lets you select one of these nodes and expand it to see other interactions. Okay, so as soon as you're into networks, you really need to have visualization to be able to work with these networks. There are a number of visualization and analysis tools. The one I recommend is Cytoscape, another of Gary's projects. It comes both in web and desktop versions, pretty easy to install locally. Once you have it, you can run, download and install networks from a variety of sources by entering their URLs. And it has a very nice ecosystem of developers who developed a series of analysis tools. I'm going to talk about gene set enrichment and other tools, and I think you've probably seen worked with Cytoscape already, so maybe I shouldn't even be saying this. But it is everything that I'm going to talk about you can do with one Cytoscape plugin or another. Okay, so let's talk about how you analyze gene sets using pathway network analysis. So there are basically three different classes of algorithm that one can apply. The most basic one is gene set enrichment, in which you have an experimental data set. And the canonical data set is a RNA expression analysis experiment in which you've, say, given a cell line a drug, and some genes have gone up, some genes have gone down, and you want to know what biological processes have been affected. And the output of this will give you a series of pathways or network groupings which are related to each other. And this is essentially the question you want to ask from this is, what biological processes have been altered in this data set? And I keep saying cancer here because this was originally written for the cancer analysis module, and I've adapted it. So wherever you see cancer, if you don't work on cancer, just think whatever your disease, replace it with whatever your disease system is or biological question you're working on. Okay, the second is de novo subnetwork construction and clustering. So here, I'm interested in discovering new pathways, new relationships among genes that are revealed by my experiment that are not in one of the pre-labeled buckets of cellular processes. And here you can ask questions like, are there new pathways in the system? Are there, differences from one cell to the next that are not apparent? Just looking at the gene lists, but when I look at them in a pathway context, I see that there is a pattern. And then the most sophisticated and advanced of these techniques is modeling. These systems take the biological pathway or the network and turn it into a computable predictive model in which you can ask what if questions. What if I knock down this gene? Or what if I ubiquinate it? What will happen to other genes in that pathway? Can I make the cell differentiate by overexpressing a gene? And these are used in a variety of applications, including drug discovery. I want to make a cell. I want to find a synthetic lethal combination of genes. I know that in this cancer, this gene has a nonsense mutation, so it's no longer active. Can I find another gene in the pathway that I can target with a small molecule that will cause an essential, which will downregulate an essential gene and cause such cell to die? So we'll go over these three techniques. Also, feel free to stop me if I'm either going too fast or too slow. So you've seen gene set enrichment, so this will be review. It's the most popular, really the dominant form of pathway and network analysis, and it has two subclasses. There's overrepresentation analysis, where you're looking for more genes in a particular process category than you would expect by chance. versus functional class scoring, where you're ranking genes according to the process that they participate in, and you're selecting the ones which are overrepresented in that functional class. These are very easy to perform analyses. There are lots of tools to choose from, and statistics are well worked out. So you can control the false discovery rate, and you can give it accurate significance testing. The disadvantages of these of fixed gene set enrichment analysis is that there are many alternative gene sets that people use. You can use any of the 3 gene ontology categories. You can use KEGG pathways. You can use reactome pathways. And in fact, there's an entire. Have you encountered MSigDB yet? No. It's a compendium at University of Santa Cruz of signaling pathways, and there are 400 or 500 different sets of gene sets that one can use. So you have to select them, and they're overlapping with each other. And one problem with treating biology as just big bags of genes is that you don't understand the relationships among those genes. So you can say that cell cycle has been altered, but exactly why it's gone up, the activity's gone up, but exactly why are you seeing that upregulation in the cell cycle? Here's an example from Reactome of the web-based pathway enrichment analysis. It's a tool that you can access from the front page. You cut and paste a list of genes, and alternatively and optionally, there some number associated with them, usually an expression value. And it will apply a gene set enrichment analysis to it and give you an interactive diagram of the cell. So this is a Voronoi plot in which it's highlighted the pathways which are overrepresented in that in your gene set, and then you can zoom down into it until you get to the pathway level, and it will show you in the colorized fashion which genes were overrepresented and why they were overrepresented. Many reactions are actually use complexes of multiple gene proteins, and so that's why you see these boxes which are half filled in, because your overrepresented gene, or the gene in your gene set you're interested in, is one member of a multi-member complex. And at the bottom of this is a list of which you probably can't read, is a list of the pathways that are overrepresented and their p-values. Okay, so that's pathway enrichment analysis, which you've already done. De novo subnetwork construction and clustering is a different kettle of fish. Here you have a list of altered macromolecules, genes, proteins, or RNAs, and you apply them to a biological network. And instead of pulling out overrepresented gene sets, it is discovering topologically unlikely configurations of those genes, proteins, or RNAs. That is, they are interacting with each other more frequently in that pathway, in that network, than you would expect to occur by chance. And so we'll then pull out those clusters of interacting genes, discard the ones which are randomly distributed, so it's removing some of the noise, and give you a series of gene interaction networks which are biologically interesting in your system. And generally, these tools will also try to annotate those clusters using gene set enrichment analysis. So they'll take, let's say you've given it 100 genes, and it finds 3 clusters of 10 genes each. It will then take each of those clusters, do a gene set enrichment analysis against a gene ontology, and try to label them. So you have a cluster that has something to do with the proteasome, a cluster that has to do with ribogenesis, and a cluster having to do with DNA replication. And then it's up to you to make sense of it. Here is I'll give you just an example from Reactome. One of our side projects is to develop a functional interaction network. This was actually created by taking the curated portion of Reactome and then carefully adding a series of high throughput functional interactions from well annotated experiments, and then tuned so our false positive rate would be less than 1%. And that's raised the coverage of Reactome from about 50% to about 61%, which is worth it. So here's an example of a very early experiment that we did with this to show the utility of the functional interaction network. So first of all, we did we took our curated, just to recapitulate, we took the curated pathways, we added a bunch of uncurated interaction networks, and then using machine learning, developed the functional interaction network with about 270,000 interactions in it. Okay, and then on a series of data sets, including a breast cancer data set, we then use this to do subnetwork extraction and clustering. So here is an example from breast cancer in which we pulled out a series of subnetworks, all of which have correspond to pathways known to be activated in breast cancer. Then we annotated those modules. And this workflow is a plugin for Cytoscape that you can get if you want it. You can look for Reactome FI Network plugin. And so the next thing that we did to show the utility is we discovered a biomarker for breast cancer survival. So we looked at each of those subnetworks one at a time, and we divided, and with a data set of a few hundred breast cancer patients, we divided them into 2 groups, one in which the average expression level of genes in that subnetwork were high, versus patients in which the average expression level was low. And sure enough, when the expression is high, these patients had a much worse survival curve. Everybody here familiar with Kaplan-Meier curves? Yeah, okay, good, very good. And the interesting thing about this is it actually was able to partition estrogen receptor positive breast cancers very well. Patients who have high ER positive patients generally have a much better prognosis than those who are ER negative, or particularly triple negative groups. And patients who have a high expression of this submodule have pretty much the same prognosis as patients with triple negative breast cancers. So it would be actually a useful clinical tool. And the advantage of doing this on a subnetwork is we also have an explanation for why this is a good biomarker. If you look at the annotation of this submodule, the annotation is cell cycle M phase and aurora kinase B signaling, both of which are markers for mitotic activity, which confirms what we know about breast cancers, which the higher the mitotic activity, the worse the prognosis, which is not always the rule in cancers. So that's just one of many different network extraction and clustering algorithms. GeneMania offers a very nice interactive version of this. There are also another popular algorithm is from Ben Raphael, who's now at Princeton, called HotNet. There's another Cytoscape app called plugin called Hyper Modules, which is particularly well suited for combining for identifying biomarkers that predict clinical characteristics in patients. And there's the Reactome Cytoscape app as well.